In lecture 4, we are going to be looking at membrane transport, which is basically going to explain how things can cross the cell membrane, how it goes in, how it goes out, what controls the direction of movement, and the various ways that these materials can cross the cell membrane. So let's start with the membrane structure and function. We know that the plasma membrane or the cell membrane is selectively permeable. Okay, it only allows certain things to cross it, and that control mechanism has to lie with the kind of proteins you have embedded within this membrane. So in addition to the proteins that you will find in the membrane, there are also going to be some lipids, right? We know that there is a phospholipids that makes up the cell membrane. There are other lipids such as cholesterol. There are also going to be some carbohydrates, which are shown here in these blue chains over here. So the phospholipids is the major component of the cell membrane. It forms the phospholipid bilayer, and by now we talked about how the phospholipid bilayer has the hydrophobic heads facing either the outside of the cell or the inside of the cell, and in between there are going to be these hydrophobic tails. The cholesterols are important because they maintain the right fluidity for the membrane. Okay, by having some cholesterol in the cell membrane, you're going to prevent it from being too solid. Okay, if cell membrane is too solid, then nothing can cross through. Right, it's kind of like a block of lard, right? It's very hard to pass through something that's a semi-solid, and you cannot have too much cholesterol in it either. High percentage of cholesterol in the cell membrane, it's actually going to make the membrane too fluid, okay, and in that case, it's going to be too liquid, and things just kind of leaked all over the place. Next, we have something called glycoproteins or glycolipids. These are proteins with sugar stuck on them, or lipids with sugar stuck on them. Okay, so over here we can see a glycoprotein. These blue blobs are the proteins with the sugar chain on them. Here is a phospholipid head with some sugar on it, so we call that glycolipids. There are different functions for these glycoproteins and glycolipids. One function is that they give the cell an identity. Okay, it's like a ID tag that the cells have, and your body can check these ID tags to see whether or not the cells belongs to the body or whether it's like an invading cell, like a bacteria. In this diagram, you can also see that there are a lot of membrane proteins. Okay, some proteins spans the entire cell membrane. Others are just kind of attaching on the sides. And so it's good to take a moment and learn about the various functions associated with the membrane proteins. So they're all summarized here, and we will be looking at each of these in more details in the slides to follow. So, as I mentioned, some proteins are going to be going through the phospholipid bilayer. It's going to span the entire length of the phospholipid bilayer. These proteins are called integral proteins. On the other side, on the other hand, rather, we have these things called peripheral proteins. So peripheral proteins are only attached either on the outside or attached on the inside of the phospholipid bilayer. Okay, they do not span the entire phospholipids. And it turns out that these proteins, depending on whether they span or not span, they will have different functions. So let's start with the first function of the membrane proteins, and that is to provide attachment sites to the cytoskeleton. In the previous lecture, we learned about there are these fibers that forms the cytoskeleton that helps reinforce the shape of the cell and provides attachment sites for organelles. And so these cytoskeletons actually needs to have anchorage point, and that's what the membrane proteins allow them to do. The actin fibers and the intermediate filaments are going to be able to anchor to the cell membrane because of the presence of membrane proteins. Okay, so you remember intermediate filaments, they form these tight junctions between two cells, so they essentially will glue the cell membranes together, and in order for them to be able to do that, they need to have attachment sites, and a lot of times these attachment sites are going to be found as membrane proteins. So in this picture, you can see the typical red blood cell shape, right? The indentation in the center, and in order to maintain that indentation, the actin fibers inside the cell must be pulling on the membrane proteins found on the surface of the cell. So that's the first function of membrane proteins. The next function we're going to talk about is cell signaling. There are some membrane proteins that are going to be functioning as receptors, and the receptors are typically going to be integral proteins. And these receptor proteins are going to have a binding site that will allow molecules of a certain shape to bind to it. Okay? And again, there is this key and lock relationship between the receptor and their targets. And once the appropriate molecule binds to the target, it's going to trigger a series of events inside the cell that will result in some kind of changes in terms of the cell behavior. And an example of that would be adrenaline. Okay, so adrenaline is something that is released in the body when you are in danger, or when you're excited, or when you're stressed out. And these adrenaline molecules will bind to a receptor on the surface of the liver cell. Okay, once the adrenaline binds to it, it will trigger a series of events to take place inside the cell, and these series of events are what we call a signal transduction pathway. And ultimately, these reactions is going to result in the hydrolysis of glycogen, okay, where you will break down glycogen and release glucose for energy, okay, which is something that you would need, right, when your body is releasing adrenaline. Let's say you are going to be running a race, you're going to need that extra glucose. So by having adrenaline in your system, you will be able to release that extra glucose. So the signal molecule never really went into the cell. They just kind of, bind to the receptor on the surface, and then all these things are going to happen inside the cell. It's kind of like Halloween. Remind me of when I was little. My parents never liked the idea of handing out candies to strangers. So what happens is we would turn off all the lights, and then if someone rings the doorbell, okay, someone rings the doorbell, like the airport, never ringing the doorbell here, then my parents would be like, go hide in the basement, okay, make no sound, so that they don't know someone is home. So the person never really got into our house, but by ringing the doorbell, it causes all these commotions inside the house. And that's kind of how the membrane proteins here is functioning. It provides kind of like an interface for molecules outside of the cells to create some kind of changes inside the cell. Here is another example. These are neurons, and neurons are found in your nervous system, right? In your brain, in your spinal cord, for example, and they need to communicate with each other. So when one neuron needs to send signal to another neuron, they would do so at the synapse. That's where the two neurons are going to be very close to each other, and they will release these things called neurotransmitters. Okay, so here the red squares are the neurotransmitters, and in order for the signal to be received by the next neuron, they must have the correct membrane protein. They must have the correct receptor for them. So once the neurotransmitter binds to the appropriate membrane protein on the receiving neuron, that will trigger a series of cellular response, which then allows the signal to be propagated. So those are the two examples of how membrane proteins can be functioning as a receptor. The next function we're going to talk about is cell signaling. There are some membrane proteins that are going to be functioning as receptors, and the receptors are typically going to be integral proteins. And these receptor proteins are going to have a binding site that will allow molecules of a certain shape to bind to it. Okay? And again, there is this key and lock relationship between the receptor and their targets. And once the appropriate molecule binds to the target, it's going to trigger a series of events inside the cell that will result in some kind of changes in terms of the cell behavior. And an example of that would be adrenaline. Okay, so adrenaline is something that is released in the body when you are in danger, or when you're excited, or when you're stressed out. And these adrenaline molecules will bind to a receptor on the surface of the liver cell. Okay, once the adrenaline binds to it, it will trigger a series of events to take place inside the cell. And these series of events are what we call a signal transduction pathway. And ultimately, these reactions is going to result in the hydrolysis of glycogen, okay, where you will break down glycogen and release glucose for energy, okay, which is something that you would need, right, when your body is releasing adrenaline. Let's say you are going to be running a race, you're going to need that extra glucose. So by having adrenaline in your system, you will be able to release that extra glucose. So the signal molecule never really went into the cell. They just kind of, bind to the receptor on the surface, and then all these things are going to happen inside the cell. Kind of like Halloween remind me of when I was little. My parents never liked the idea of handing out candies to strangers. So what happens is we would turn off all the lights, and then if someone rings the doorbell, okay, someone rings the doorbell, like the epinephrine ringing the doorbell here, then my parents would be like, go hide in the basement, okay, make no sound, so that they don't know someone is home. So the person never really got into our house, but by ringing the doorbell, it causes all these commotions inside the house. And that's kind of how the membrane proteins here is functioning. It provides kind of like an interface for molecules outside of the cells to create some kind of changes inside the cell. Here is another example. These are neurons, and neurons are found in your nervous system, right? In your brain, in your spinal cord, for example, and they need to communicate with each other. So when one neuron needs to send signal to another neuron, they would do so at the synapse. That's where the two neurons are going to be very close to each other, and they will release these things called neurotransmitters. Okay, so here the red squares are the neurotransmitters, and in order for the signal to be received by the next neuron, they must have the correct membrane protein. They must have the correct receptor for them. So once the neurotransmitter binds to the appropriate membrane protein on the receiving neuron, that will trigger a series of cellular response, which then allows the signal to be propagated. So those are the two examples of how membrane proteins can be functioning as a receptor. Next, we have enzymatic activities. Enzymes, as you learned in lecture 2, are proteins that catalyze chemical reactions. They allow chemical reactions to take place faster by providing an active site for the substrate to bind into. Substrate is the thing that the enzyme is going to be working on, right? And again, there is a key and lock relationship between the substrate and the active site of the enzyme. So this one will only allow circles to bind to it. This one will only allow triangle to bind to it. Okay. Sometimes you need to do a bunch of chemical reactions very rapidly near the surface of a cell. Something that will happen, for example, in your immune system. There is something called the complement proteins. That's part of the immune defense that you have, and you need to activate multiple chemicals in tandems, one after the other, and you have to do it quite quickly near the surface of the cell sometimes. And so what happens is you will form almost like an assembly line where the product of one enzyme, so the circle goes into enzyme 1 and splits out the product, which is a triangle, will now go into a second enzyme, and then it will quickly process it into something else, and then it will go into the next enzyme, and then the next enzyme. So by having the enzymes arranged in close proximity near the cell membrane, you are able to catalyze the reaction very, very quickly in a domino fashion almost. The next one is intercellular junction. We briefly mentioned about this earlier already, but there are some specialized proteins located in the membranes that will allow them to kind of hooked on to the cells that are next to it. Okay, and of course they cannot do this by themselves. They would also be working in conjunction with the intermediate filament to form these tight junctions, and that is important to prevent uncontrolled movement of materials between cells. Another function for membrane proteins is what's called cell-cell recognition. You have trillions of cells in the body, so how does your immune system know what belongs to the body and what does not? Well, earlier we talked about the presence of glycoproteins and glycolipids that are found on the surface of the cell. So these glycoproteins are sugary proteins and sugary lipids that will serve as an identification tag for your immune system to use so that they can distinguish self versus non-self. So here is an example of one of your immune cells called the natural killer cell, and it's basically trying to attack this foreign cells because it doesn't have the right ID tag, right? The glycoproteins and glycolipids that is on this foreign cells does not match the ones in your body, and so your immune cells will be able to identify it, and then they are going to attack it. So if this system is messed up, it could end in one of two ways. Number one, you might not be able to mount an immune defense against a bacteria or virus, and of course that could cause you to become very ill, or in another way, if your immune cells does not distinguish between self and non-self, they might end up attacking your own cells, and that could result in autoimmune diseases where your immune system attack your own body. And examples of that would be things like ALS or MS, multiple sclerosis, lupus. All these are examples of autoimmune diseases where the immune cells fail to tell apart what belongs to the body and what does not. Last but not least, membrane proteins are also going to be involved in transport. That is, they are the ones that are responsible for controlling what can go into the cell and what's going to come out. They are going to regulate the movement of materials across the plasma membrane. So here you can see a transport protein grabbing onto one of these circles, and then by changing the shape like a revolving door, it's going to split the circle onto the inside of the cell. Before we talk more about membrane transport, we should spend a few moments discussing the concept of cellular energy. First, metabolism. Right? You hear people say, I have high metabolism, low metabolism. Exactly what does it mean? Metabolism basically refers to all the chemical reactions that are going to be taking place inside a cell. Okay, and there are two types of metabolism. The first one is catabolism. Okay, catabolism refers to reactions that are going to be breaking down molecules into smaller units, and that tends to release energy. Okay, so think about catabolism. The root word here, the prefix, is same root word as catastrophe. In catastrophe, everything breaks down, right? So that's how you can remember it. Catabolism involves breaking things down. Okay, so you might be breaking down polymers into monomers, and that will in turn release energy. On the other hand, we have anabolism. Okay, and anabolism is the exact opposite of catabolism. Over here, you are going to be constructing, you're going to be building molecules from smaller units. So you might be taking monomers and you will be linking them to produce polymers, and to build things that requires energy. So the energy released by the catabolism is often used to drive the energy required for anabolism. So here is a better view to show you the relationship between the two type of metabolisms. Okay, you're gonna eat your food, and in your food you're gonna have all sorts of macromolecules, right? You have your carbohydrates, you have your lipids and proteins and whatnot. Those are going to be rich in building blocks, in monomers. So in catabolism, catabolic pathways, you're going to be breaking those molecules down into their building blocks, into their monomers. So you will be breaking your proteins into amino acids, you'll be breaking your lipids into glycerol and fatty acids, and as you're doing these kind of catabolic reaction, you are going to be releasing a useful form of energy. Okay, and as a kind of side effect, you're going to be generating heat as well. We are kind of like a light bulb, right? You touch a traditional incandescent light bulb, it's going to feel hot because a lot of energy is wasted as heat. We are the same way. Okay, we're not very efficient in converting food into energy, and so much of it is going to be lost as heat. And that's not all a bad thing, because the heat is used to maintain our internal body temperature of 37 degrees Celsius. So it's not entirely bad. Okay, and then you're gonna take these building blocks, and then you're gonna build something that you need in the body. Okay, so for example, actin is a protein that we need, right? So you will take the amino acid that's necessary to build the actin, and you are actually going to build them using the anabolic pathways. Okay, so these are your anabolism, and to build things, you are going to need energy. So the energy that you release from your catabolic reactions are going to be used to drive the anabolic reactions. So cellular respiration, which we have been talking about for quite some time now, is the process of taking glucose, reacting it with oxygen to produce carbon dioxide, water, as well as usable energy. So in the context of what we're discussing right now, cellular respiration is actually a type of catabolic reaction where you will be breaking down glucose into carbon dioxide, and in the process the energy that are released will be captured and stored in the form of what we call ATP. Okay, so here is an overview of cellular respiration. We are actually going to go into all the details in lecture 5, but suffice to say for now that it generates quite a bit of ATP, the usable form of energy, and we can then use these ATP to do different things. So this is what ATP looks like. ATP stands for Adenosine Triphosphate. So adenosine is derived from one of the nucleotides that we learned, the adenine, and you can see that there is the ribose sugar here, and there is the adenine, and it's going to be connected to 3 phosphate group. Okay, that's why it says triphosphate, right? Like a triangle has 3 sides, a tricycle has 3 wheels. Triphosphate means you have 3 phosphate groups attached to the molecule. So this is the ATP molecule, and ATP molecules represent a fully charged molecule. Okay, there's a lot of energy that is contained in this molecule, and it's ready to be used. When you want to extract that energy, when you want to release that energy for whatever use, you will need to break the ATP by taking off one of the phosphate groups at the end. So when you add water molecule to your ATP, you are basically doing a hydrolysis reaction that will convert it into a phosphate group, as well as what's called ADP now. Okay, so you see that one of the phosphate group has been broken off, so it only has 2 phosphates now, and that's called diphosphate. So ADP is the empty uncharged version of the molecule, and the ATP is the charged version. More importantly, when you break the ATP into ADP, you will be releasing energy, and that energy can be used for various things. You can use it to build molecules, right, in the anabolic reactions, for example. So here is the cycling reaction between the two different forms of the molecule. You can use the energy you create from the catabolic reactions, say by breaking down glucose in cellular respiration. That releases the energy to charge up the ADP. Okay, so that energy will allow you to add the phosphate group to the ADP and fully charging it in the form of ATP. So the ATP will hang around in the body until you need it for something. Then to release the energy, to harness the energy from the ATP, you will need to add a water molecule. So the water molecule will hydrolyze the ATP and converting it back into ADP and phosphate once again, but in the process you will be releasing energy, and these energy can be used for anabolic reactions. In addition to anabolic reactions, we can actually use ATP to power all sorts of cellular activity. For example, we can use it to power mechanical work where you have to physically move some parts of the cell, right? So moving a flagellum, for example, moving a cilia, for example, that requires ATP. And again, to get that energy that's locked within the molecule ATP, you need to hydrolyze it by adding water molecule. And then once it's used up, it will get converted into ADP and P. So this ADP and this phosphate group will need to be recharged, right? And in order to recharge it, you would need to get energy from, say, burning glucose or burning other food that you've eaten. We also can use ATP for transport work. When we move things across the cell membrane, sometimes it requires energy. It's like, you know, moving a box, right? Moving it uphill is going to require energy. Moving it downhill, you just kind of let it go, right? Which is gonna slide down the slope on its own because of gravity. So similar concept occurs in the context of transport across the cell membrane. Some transport is going to require energy in the form of ATP, others not so much. Another way we can use ATP is to do chemical work. You might remember enzymes will catalyze chemical reactions by lowering the activation energy, right? So something has to compensate for that energy difference, and in some cases, the ATP molecule is what we need to adjust for the energy requirement difference. So this is probably a good place to stop for this video. We will continue to talk about the different types of transport in the next video. Thank you for watching.